So just here, first part of the talk, the proximity labeling and NLR receptor was carried out by Emilian Zhang, Nago, and others helped in this project. It was a collaboration between Iowa State and Stanford. Top 3 project was primarily carried out by Jiang Chen Wu with the help of Insuk Park. And there are lots of collaborators on this project from Seoul National University on Magna Porta, Texas A&M on sclerotonia. Unfortunately, Marty Dickman passed away last year. And then other pathogen work was done with collaboration with other people at UC Davis. Most of the work I presented today was funded by NIH, NSF projects, and the other projects in the lab which is funded by this. So we are grateful for funding agencies actually. So thank you for your attention, and hopefully I gave you a little bit about what's happening in our group currently.
Thank you, Dr. for an excellent presentation. I'm the chairman of the system.
Thank you, sir, for excellent presentation on the topic of the emerging perspectives on plant immunity. You have covered a lot on your work with respect to the tobacco virus, the virus-induced gene silencing, and also autophagy. Especially, it is an emerging area, especially on autophagy with small molecules. I feel we can take a long way in doing with this and screening of small molecules for regulation. Thank you, sir, for excellent presentation. Sir, Chairman, sir, can you take 1 or 2 quick questions? Sir, it is muted, sir. One or two quick questions, if anybody has got.
Yes. Hi, this is Sridhar here. Can we ask a question over the phone or is it on the chat?
You can just ask.
Huh?
I can hear you.
Sir, I have one question.
Yes. Hello.
Hear me?
Dinesh, is this clear?
What is the question? Yeah.
I would like to know how easy it is to label proteins in vivo, understand protein-protein interaction.
What was the question? To label proteins in vivo?
How to label biotin labeling of the proteins in vivo to understand protein-protein interaction.
Yeah, so you just you can just inject the biotin into the tissue, like for aggregation. You don't need to do any special move, actually. You just have to infiltrate the biotin solution into the tissue, so just like what you do for radioactivity.
Yes. Sir, in the chat box, there is a question. Shall I read, sir?
Yeah.
Does URB3 triggers against RNA viruses, or has it showed any interaction with DNA viruses?
So far, we have only tested for the RNA virus, but the current testing not DNA virus, but also pathogens like bacterial and fungal pathogens. Actually. So we currently don't have the data, but we have ongoing experiments to test those things. Yeah.
Sir, I have one question.
Yeah.
Virus will produce the gene silencing silencer. How to manipulate that gene silencing silencer in the host system?
I mean, there are a lot of different ways to do it, but you know, the problem with that is that the host also needs the these RNA silencing suppressor system. So often what happens is these proteins like dicer-like proteins, RNA polymerase, those are the ones which monitor RNA silencing suppressors. So we could work with those proteins that to manipulate RNA silencing suppressors from the viral side.
Thank you, sir.
This is the last question. We have one more in the chat box from Nigesh. Can we use H4 for control of botrytis infection in castor?
In last part, what was the last part of?
Can we use H4 for the control of botrytis infection in castor?
We have not tested it, but everything we have tested botrytis so far seemed to work, I think. Yeah. So one could test it. So we are right now submitting this paper. So yeah.
Thank you, sir, for your excellent presentation. I once again thank on behalf of the organizing committee, on behalf of the chairman, and my personal behalf for sharing your valuable experiences with respect to the triggered immunity of the latest happenings. Chairman, sir, we'll go with the next speaker.
Thank you, Dinesh sir. Dr. sir.
Yeah.
Jagdish Gupta, sir, are you there?
Yes. I'm.
Yes, sir.
Now sharing the.
Sir should be. Hello, screen ತೆಗಿಬೇಕು. Dinesh sir.
Please, can you please close your screen, sir?
Yeah.
Okay.
Yeah, thank you very much. To you.
So title of my talk is the role of nitrate nutrition and nitrate signaling in plant pathogen interactions. Okay. So before going to that, I'm going to give a small introduction.
Sir, Dr. Jagdish Gupta.
Yes.
I have the privilege to introduce to the delegates for a few minutes, sir. I will take only 2 minutes.
Sure.
I welcome you on this platform.
Camera, sir.
Nitrogen nutrition and nitric oxide signaling in plant pathogens interactions. I have the privilege to introduce Dr. Jagdish Gupta Kapagundi. He is worked as a staff scientist at the National Institute of Plant Genomics from 2014 to 2018. And he was a Madame Curie Fellow at the University of Oxford, UK from 2012 to 2014. Then scientific staff at the University of Rostock, Germany from 2010 to 2012. He also worked as a postdoctoral fellow at the prestigious Max Planck Institute of Molecular Plant Physiology, Germany, from November 2007 to 2010. His contributions in the field of plant molecular interactions have brought several honors and recognitions. To quote few of them, he was the International Scientific Committee member. He is presently a member in 8th Plant Nitric Oxide Symposia in 2021. 2020, he is working as Associate Editor of Food and Energy Security. In the same year, now in this year, presently, he is editorial review board of the Plant Cell and Environment, editor of Plant. Then he is the prestigious award winner of Bayrak Big Ignition Grant Award. He is a member of several societies like Society of Redox Biology and Medicine, then Madame Curie Alumni Association. He gave several talks. He was the inaugural talk in the Post-Genomic Technologies Conference in Russia. His contributions have recognized both in India and abroad. And he is the co-organizer of India EMBO Symposium. And he is worked as a Russian Science Foundation pool of international experts in 2019. And he was visiting Max Planck Institute, Germany from March 2019. And he is a prestigious DST DAD fellow from 2018 to 2020. And all these contributions are recognized with his highest H index and the citations. You believe it or not, the highest H index of 31 with more than 4,000 citations for his scientific contributions. They speak the contribution of Dr. Jagdish Gupta. Now I invite Dr. Jagdish Gupta to deliver his talk on the role of nitrogen nutrition, nitric oxide signaling in plant pathogen interactions. The floor is yours, sir. The time allotted is 30 minutes. After 25 minutes, you will be given a signal like this, 5 minutes, and then we have 2 minutes discussion. Thank you.
Thank you very much for a very nice introduction. So, the title of my talk is the role of nitrate nutrition and nitric oxide signaling in plant pathogen interactions. Actually, there is some technical issue. I have to share it again. Okay. Before going to that, I'm going to give an introduction about the importance of nitrogen in plants. So we know the nitrogen is essential for the growth and development of plants. Why it's so important is that it plays very important role in biosynthesis of chlorophyll, nucleic acids, amino acids, as well as vast number of metabolites. So, N deficiency occurs in the plant due to slow metabolization and lack of sufficient organic matter, and leaching due to heavy rainfall and increased activity of denitrifying bacteria, which can deplete nitrate content and nitrate content in the soil. And the consequences of the low nitrogen is that low nitrogen can retard the plant growth and cause severe physiological and morphological defects. Then, how do plants cope up with the low nitrogen? So the plants have evolved N-uptake systems, nitrogen uptake systems, to support their growth and survival under nitrogen deficiency. So the plants have two kind of affinity transport systems. One is low affinity nitrate transport system. Second one is high affinity nitrate transport system. So under high nitrate concentration in the soil, what happens is that there is a production of nitric oxide. The produced nitric oxide causes nitrogenation of high affinity nitrate transport system. Therefore, hats do not work under high nitrate concentration. That leads to operation of low affinity nitrate transport system. But when the nitrogen concentration is low, the NO concentration is low, and then the reduced levels of NO, some minimal amount of NO, it activates the thioredoxin, which can cause the denitrosylation of the high affinity nitrogen systems that leads to operation of the high affinity nitrogen systems. So nitrogen plays very significant role in plant defense against both biotic and abiotic, in virulent and virulent pathogens. For example, the various papers showing that nitrogen play very important role in plant resistance, and recently we have also shown that form of nitric nutrition affects the resistance against Pseudomonas. Salicylic acid and defense signaling molecules such as nitric oxide depends on the nitrogen. Then how NO is produced in the plants. The plants have various pathways that produce nitric oxide. Recently, we divided them into two categories, reductive pathways and also oxidative pathways. The important reductive pathway is nitrate reductase. This is present in the cytosol. It uses nitrate. It converts nitrate to nitrite. The produced nitrate becomes a substrate for the mitochondria to produce nitric oxide. So plants also have arginine dependent pathway, like nitric oxide synthase like activity, but there is no protein origin has been identified for this pathway. So the main predominant pathway is nitrate reductase. Then the produced nitric oxide, how it acts as a signal is that the produced nitric oxide acts as a signal by a process called nitrosylation. Nitrosylation is reversible binding of NO to the thiols of the reduced-cysteine residue. So once the protein become nitrosylated, it becomes active or inactive. One of the very important example is that NPR1. NPR1 present in the oligomeric form in the cytosol. Upon nitrosylation, the NPR1 become monomer. The monomers move into the nucleus where they interact with the TGA1 transcription factors that leads to activation of the PR genes. So, since the NO production also requires nitrate, it may play important role in the elicitation of the local acquired resistance, where the resistance is developed at the local sites, and also systemic acquired resistance, which results in the broad spectrum disease resistance against various against secondary infection followed by the primary infection.